When Charles Darwin first published On the Origin of Species, he admitted that the sheer complexity of the eye appeared at first glance nearly impossible to explain through gradual evolution. Yet discoveries in genetics and anatomy confirmed the vertebrate eye is a masterpiece of cumulative adaptation built over hundreds of millions of years. This phylogenetic mapping demonstrates that vision emerged through the repeated modification and repurposing of ancient photoreceptive systems inherited from the earliest metazoans. Mapping the genetic lineage explains how the eye changed, but it leaves a gap regarding the underlying physics of the design. Out of countless theoretically possible biological architectures, why did evolution converge so consistently on this specific design, while apparently ignoring thousands of other solutions? To find the answer, we have to look past the anatomy and into the Brain Kinematics Model, or BKM, and continuity theory. This perspective treats sensory organs as active biological translators, sitting at the intersection of the external environment and an organism's internal organizational dynamics. Evolutionary history is the process of optimizing the fidelity of translation between environmental strain fields and the internal organizational continua of the brain. Under the BKM framework, the history of the eye is the history of information processing, where physical anatomy serves primarily as a vehicle for translation. The external environment consists of continuous physical fields, electromagnetic radiation, mechanical forces, and chemical gradients. Biological information begins at the exact moment a continuous physical wave from the environment strikes a biological boundary. Certain biological molecules, like opsins, naturally undergo conformational changes after absorbing photons, effectively turning physics into chemistry. In the language of BKM, this translated data is called a strain package. A strain package is an organized physical perturbation that has been translated into an internal state capable of influencing future biological dynamics. Light provides a high information-to-energy ratio, revealing distant predators and spatial orientation with minimal energy expenditure. Primitive cells used these initial strain packages to regulate basic survival behaviors, such as moving toward light for photosynthesis or away from harmful UV radiation. These early photoreceptors acted as simple transducers, converting environmental strain into a raw internal signal to predict impending changes in the surroundings. Biological systems rarely invent new physics from scratch. Instead, they modify what they already have. This is known as evolutionary co-option, the reuse of existing molecular tools for new functions. We trace the origin of photosensitivity back to ancient GPCR cascades. Originally used for basic chemical sensing, they were modified to interact with light. This repurposed toolkit became the foundational architecture for vision across divergent species. Because the system was successful and deeply integrated into early life, historical contingency locked future evolution into modifying this specific genetic framework. The modern eye's architecture is a testament to this co-option, showing that sensory systems are built from existing internal communication networks. However, the sensory gate, the brain, and the resulting behavior function as a single, inseparable loop. Anatomical improvements in a receptor provide no benefit without a parallel increase in the neural capacity to interpret the incoming strain packages. Continuity theory describes this interaction as countercurrent translation. Physical movement and behavior continuously alter the environmental strain field hitting the receptor, which in turn updates the internal organizational state. Predatory dynamics demand rapid estimation of velocity and future position, which is why the retinae evolved to perform complex pre-processing before a signal ever reaches the brain. The eye co-evolved in tandem with the brain and motor systems. It is an integrated component of an information processing loop. If mutations continuously present new biological options, why has the GPCR-based design remained stable for hundreds of millions of years? Sensory evolution must navigate rigid physical constraints, from thermal noise to the energy costs of high sensitivity. These constraints suggest a principle found in engineering, impedance matching. Evolution favors receptors that couple the external physical field with the organism's internal biochemistry with high efficiency. Effective coupling preserves the organization of the incoming signal, while a mismatched receptor degrades the information during the transition from physics to biochemistry. The specific molecular structure of the opsin receptor matches the energy yield of a photon, allowing for precise information transfer. Natural selection favors this robust informational coupling over designs that might offer more sensitivity at the cost of stability or energy. Evolution works to stabilize these high-fidelity translators because they provide the most reliable map of the external world. This need for optimal energy information coupling across biological boundaries drives the emergence of sensory systems. This translational logic extends beyond light to every physical interface an organism maintains. Whether it is a mechanical wave of pressure, a thermal gradient, or a chemical concentration, each is translated into the same common internal format. These varied inputs converge within the internal organizational continuum. Different physical continua are transformed into comparable internal strain packages, creating a unified informational language for the nervous system. Memory, perception, and even symbolic cognition are the result of the recursive organization of these translated packages. The Brain Kinematics Model suggests that evolution is the progressive, continuous refinement of translation between the external universe and internal biology. By identifying these mathematical and physical constraints, we can see vision not just as a biological accident, but as an inevitable result of how life organizes information.